Good morning, dear student. In previous lecture, we discussed different types of structure of proteins, that is primary, secondary, tertiary, and quaternary. So, in today's lecture, we are very much interested in the concept that is the denaturation of protein. So, what is this denaturation of protein? Again, it is very important concept. So, you should know what is denaturation of protein and what are the denaturating agents which causes the denaturation of protein. The word itself indicate denaturation means there is a disintegration of the native form of the protein. So you should remember in denaturation process or in the denaturation of protein, there is a conversion of functional protein into non-functional form. So functional protein, it is converted into non-functional protein, then it is considered as the denaturation of protein. In other word, we can define this denaturation. So denaturation may be defined as the disintegration. So we see the definition of this denaturation. It is defined as disintegration. It is defined as disintegration of functional protein of functional protein. So the functional protein may be your secondary, tertiary, and quaternary. So this is your functional protein. So it is defined as disintegration of functional protein, that is disintegration of secondary, tertiary, and quaternary structure. Disintegration of functional protein, that is secondary, tertiary, and quaternary structure of the protein. So what is this denaturation? It is defined as disintegration of functional protein, that is disintegration of secondary, tertiary, and quaternary structure of the protein. Okay, and another important point is that due to this disintegration, uh, there is a alternation alternation of physical chemical alternation of physical, chemical, and biological characteristics characteristics of the protein by variety of agent by variety of agent. So let us discuss once again what is this denaturation. Denaturation is nothing but the conversion of functional protein into non-functional form, or we can say that the complex structure of the protein, it is converted into its simplest form. Again, if you see its definition, denaturation of protein is defined as disintegration of functional form of the protein, that is conversion of secondary, tertiary, and quaternary structure of the protein into its simplest form. Okay, and there is alternation due to this denaturation process. There is alternation of physical, chemical, and biological characteristics of the protein by a variety of agents, by a variety of different denaturating agents. Again, if you consider the native proteins are said to be the proteins occur in plants and animal tissue. So we know that whatever the functional protein, it is considered as the native form of the protein, and these proteins are occur in animal and plant, and they possesses different characteristic properties such as solubility. So if you consider the third important point, that is the native form of the protein. Native form of protein. Native form means the functional form of the protein, or the native protein. It is considered the native form of the protein. It is found in plants and animals, plant and animal tissue, plant and animal, plant and animal tissue. So, so whatever the protein which is found in plant and animal tissue, it is considered as a native form of the protein, and they possesses this native form of the protein possesses different properties such as solubility, viscosity, optical rotation, sedimentation rate, and electrophoretic mobility. So this native form of the protein possesses different properties such as solubility, or different characteristics such as solubility, then viscosity. So these are the properties of this native form of the protein. Viscosity is there, then optical rotation is there, optical rotation is there, and then a sedimentation rate is there, and electrophoretic mobility is there. And due to this denaturation process, due to this disintegration of this native form of the protein, there is a change in this property, that is its solubility, viscosity, optical rotation, electrophoretic mobility, and sedimentation rate. So let us discuss these denaturing agents, the agent which is responsible for denaturation of the protein. So this is very important concept. You should remember that the conversion of functional protein, or the native form of the protein, into its simplest form, that is conversion of secondary, tertiary, and quaternary structure of the protein into a simple form, it is called denaturation of protein. And due to this denaturation, different physical, chemical, and biological characteristics of the protein may be changed by a variety of agents. And the native form of the protein, which is found in plants and animals, with having different physical properties, and these properties can be changed by this denaturation of the protein. So let us discuss the denaturing agents. The denaturing agents. The important point is denaturing denaturing agents. Denaturing agents in denaturation process of the protein. The first one, that is your physical agents. Physical agents. If you consider the physical agents such as heat, then surface action, surface action, then UV light, ultraviolet light is also there, UV light, then ultrasound also there, ultrasound, and then the high pressures. So these are the some physical agent which is responsible for the denaturation of protein, that is heat, surface action, UV light, then ultrasound, and high pressure is there. Again, the second one, that is your chemical agent. Chemical agents. Chemical agents, generally the acids, alkali, heavy metals, urea, ethanol, urea, ethanol, guanidine, it is a base, guanidine, GUAN. Okay, I will make a mistake. Guanidine, that is GUANIDINE. Guanidine is also the nitrogen base and detergent, and the last one is detergent, etc. So these are the chemical agent which is responsible for the denaturation of the protein, conversion of the native form of the protein into its simplest form. Again, mainly this urea and guanidine are probably the interfere with hydrogen bond between the peptide linkage. So this urea and this guanidine, so they are interfere, if you consider, they are interfere in the hydrogen bond between peptide linkage, interfere with hydrogen bond of peptide linkage, peptide linkage. So whatever the hydrogen bond is there uh, between the two peptide linkage, two peptide chain, and then this urea and guanidine, it will interact with this polypeptide chain and the bond is break, and that is nothing but the denaturation. So this urea and guanidine, it is a nitrogen containing component, interfere with hydrogen bond present in peptide linkage. While acid and alkali, if you consider this acid and alkali, they are directly attack the hydrogen bond in the secondary and tertiary structure of the protein. So this acid, so here we can write acid and alkali, acid and alkali, that probably attack directly the hydrogen bond, directly the hydrogen bond in the secondary, in the secondary and tertiary structure of the protein, and tertiary structure of protein, tertiary structure of protein. So you should know which are the physical agent, which are the chemical agents. So physical agents such as heat, surface action, UV light, ultrasound, high pressure is there. Chemical agents such as acid, alkali means base, heavy metals, urea, ethanol, guanidine, detergent, etc. So this urea and guanidine, it is interfere with hydrogen bond of peptide linkage, while this acid and alkali directly affects the hydrogen bond in the secondary and tertiary structure of the protein. So if you consider uh, what is the changes, that is physical, chemical and biological changes, so let us discuss the change or alternation. So let's discuss the first one, that is physical alternation. Physical alternation. Physical alternation. So many proteins, especially the globular type of protein, can be crystallized in its native form. So you should remember the first important point is that many proteins which is globular type, so globular type of a protein can be crystallized in its native state, so can be crystallized in native state, means in the functional form. So this globular type of protein can be crystallized in native state, but due to this denaturation of the protein, they cannot be crystallized. So second important point, due to this denature protein or denaturation of protein, denature protein cannot be crystallized, cannot be crystallized in native state. That is your physical alternations. So the globular type of protein that can be crystallized in native state, but the denature protein or denaturation of the protein does not results into the uh, these proteins. Due to this denaturation process, they cannot crystallize. Okay, this is the physical alternation. Again, if you consider second, that is chemical alternation. Chemical alternation, if you consider the denatured protein, it is greatly decreased in its solubility at its isoelectric point. So what is the chemical alternation? So decrease in solubility, decrease in solubility, or decrease in solubility at its isoelectric point. Isoelectric point. So at its isoelectric point. So we are very much familiar with this isoelectric point. So the chemical alternation means the decrease in solubility at its isoelectric point. Again, the chemical groups are exposed to chemical reaction. These chemical groups of proteins are exposed to chemical reaction and more readily detected as a result of unfolding process in denaturation. So at least you should know what is physical alternation. That is, the globular type of protein can be crystallized, but the denatured protein cannot crystallize. If you see the chemical alternation, the decrease in solubility at its isoelectric point. And if you see the third one, that is the biological alternation. Biological alternation, then the digestibility of certain denatured protein by proteolytic enzyme is increased. So in this case, the digestibility of certain denatured protein by proteolytic enzyme is increased, and the enzymatic or hormonal activity is usually destroyed by denaturation. So you should remember the important point. The enzymatic or or hormonal activity hormonal activity is usually destroy is usually destroy due to this denaturation of the protein. Again, the antigenic or antibody function of protein are frequently altered. The second important point is that antigenic or antibody function antigen or antibody function of protein are frequently change. Okay, again, if you consider, if the denaturation is severe, means if it is present in excess amount or in which is carried out in large amount, if the denaturation is severe, then the protein molecule becomes insoluble and precipitation results, as well as the changes in the properties of the protein are permanent and it is irreversible. So you should remember, if denaturation is severe, if it is in large amount, then the protein molecule becomes insoluble and the precipitation results. So the denatured protein undergo precipitation results, as well as the changes in the properties of protein are permanent. So whatever the changes which is occur, they are permanent and irreversible. in case of severe denaturation. But in case of mild denaturation, there is a reversible denaturation leading to slight changes in the properties of the protein, which can be restored to the native state after suitable treatment. So you should know the severe form, it is irreversible, while the mild denaturation is considered as a reversible denaturation. So severe denaturation, it is considered as irreversible denaturation, while the mild denaturation, it is considered as a reversible denaturation, which again convert, that is the denature form of the protein again convert into its original form or into its native form or into its functional form by suitable treatment. So this is all about the denaturation of the protein. In next lecture we will discuss the remaining important points of this chapter. Thank you.